Hello, everyone. I'm Mohini Patil, and it is my pleasure to welcome you all and kick off Thermo Fisher Scientific's Latin America Quick Talks in Aquaculture Genomics. Before we launch into the talk by our esteemed speakers, I'd like to share an overview of our Applied Biosystems Axiom Aquaculture portfolio. So a little bit of history on our innovations at Thermo Fisher Scientific for the Axiom microarray business. Back in 2011, we began with customized arrays, followed by the design of multi-species arrays that allowed end users to lower their costs and consolidate their inventory. With developments in our analysis pipelines, we are able to analyze polyploid species now. This was followed by the introduction of the low-cost, high-throughput Axiom 384 HT format. Further strides in our analyses pipelines have allowed us to interrogate large insertions and deletions. In 2016, with the release of our Axiom microbiome solution, customers were able to analyze virus and bacteria in a single solution. And over the past 3 or 4 years, we have been able to design complex markers with high GC content, and have also launched copy number analysis solutions. Last year, we released the Axiom Propel solution that has been transformative in lowering running as well as scale-up costs for our high throughput customers. And by 2030 now, we estimate that 62% of the food we eat will be farmed. Worldwide consumer preference for and consumption of aquatic species is increasing. Guidelines from the American Heart Association prevent the importance of eating fish to help protect heart health. This increase puts a lot of pressure on our aquaculture industry to meet the market demands. And applications of genomics in this market helps drive growth in the field by helping to improve fish production, decrease loss from disease and pests, increase traceability, and optimize feed formula for healthy, profitable, and sustainable fish farms. And we at Thermo Fisher have several tools that enable the advancement of genomics in the aquaculture market. On the left here, you can see we have a lot what we call our expert arrays, and these are public arrays available to everyone globally. These include species such as Atlantic salmon, Arctic char, catfish, coho salmon, tilapia, to name a few. And on the right, we have our custom arrays. These are only private to specific end users who design them. And these include barramundi, species such as black-lipped pearl, black tiger prawn, flounder, and lumpfish. Again, this just a taste of some of the things we have in our portfolio. And in addition to all the various species, we also have the ability to design multi-species arrays. And what does this mean? This means that we can have multi-species on a single array plate, and it helps you to consolidate your inventory, and it also helps save costs. And in addition, collaborators can work together to design a single array and yet maintain the privacy of their content. We have no restriction on the number of species on the array or the number of markers per species on the array that can be tiled. And so I mentioned our analysis capabilities that we've been constantly innovating. And this is just to highlight some further investments that we've made in our analysis software and bioinformatics. This allows you to publish faster, win awards, as well as make accurate reading decisions, as shown in the specific oyster paper by Alejandro and Ross Houston and other authors. You can also identify animals that are resistant to disease, as demonstrated by Alistair Hamilton and his authors here. So we anticipate that as the need for genomics and aquaculture increases, there will be an increase in sample volumes in the future. And in order to keep up with these demands, we launched the Axiom Propel workflow solution. And this new workflow solution, there is a lower investment cost as you scale up the sample volumes. There's reusable consumables that help minimize plastic and eliminate costly pipette tip waste, and also a faster turnaround time that helps you keep up with the breeding program demands. We have also introduced a standalone scanner and bulk reagents that enable simplification of the workflow, as well as an increase in flexibility and efficiency. And so I just want to wrap up. Just to summarize, the Axiom genotyping solution for aquaculture will allow for market consistency, flexibility in customization, so you can design arrays of your own species, and it also allows for affordable, scalable, and rapid workflow. So as we like to remember it, it's faster, flexible, affordable, scalable, tailored, and rapid. And so again, thank you everyone for attending this series, and please enjoy the upcoming talks.